are here in our link if you want to know more about the active protocols that we have in terms of research. So without delaying more this, I will invite Paola Soares-Mead to the podium. She will be talking about targeting the margin after resecting the core. I think there's no introduction needed. Paola is just amazing. She has been doing a great job in research. And welcome, Paola. Thank you so much.
Thank you.
And while we get ready for Paul, I would like to acknowledge the great attendance that we have this morning with close to 25 virtual participants from 8 different countries, including Bolivia, Colombia, Mexico, Italy, India, Thailand, Peru, and multiple states in the US. Thank you.
All right, good morning, everybody. Thank you for being here today in person and also virtually. My name is Paola Soares-Mead. I'm a postdoc research fellow at Dr. Q's laboratory. And today I'm going to talk to you. Let me see if I can move to the next one. This one's a lot better. I'm going to talk to you about a very exciting story that is currently developing in the laboratory while we're studying glioblastoma. And I hope you like it as much as I do. And the title the talk is titled Targeting the Margin After Resecting the Core, Reevaluating Spatial Heterogeneity in the Glioblastoma Tumor to Guide Treatment of Recurrence. And I want to give a special shout out before I begin to my student Rachel, because she's helping me take this to a different level. So talking about GBM, we know that it's the most common and aggressive primary malignant brain tumor in adults. It is a highly invasive tumor with a 5-year survival of less than 5%, in which gold standard treatment is still surgical resection followed by temozolomide and radiation therapy. And even despite many years of research and different efforts, we still don't have a curative treatment in 2023. One of the most important clinical challenges that we have that translates all the way back to the laboratory is that this tumor is highly invasive. And when we talk about invasion, we talk mainly about 2 biological behaviors, which is glioblastoma migration, so meaning the ability of the cells to move inside the brain, and also glioblastoma proliferation, how the cells are dividing in the brain. And here I'm showing 2 videos of this new technology that we have in the laboratory, in which there are only about 6 machines in the world. We got this in collaboration with Dr. Huang in artificial intelligence, and this technology is called TomoCube, which is basically a CT scan of the cells, but instead of using radiation, we use light. So this allows us to do labeling-free imaging of the cells, and you can see literally all the organelles of the cells and how they're moving. So when we talk a little bit more about invasion, and as you can see in the video on your right, we can see that the GBM cells use the brain side architecture as highways for migration. It's been described that these cells use the white matter tracts and the perivascular spaces to migrate. We know, especially by the work by Dr. Schiaparelli, that GBM cells modify their volume and their whole cytoskeleton for invasion. Basically, they squeeze through very narrow spaces in the central nervous system, and that they alter their surrounding extracellular matrix to make it more amenable for invasion. Even if we're doing supramarginal resections, we know that there are some cells that are still moving and sitting and staying inside the healthy brain, and those are the culprits of tumor recurrence. And we can see that here. There's a hypothesis that highly migratory cells are responsible for this tumor progression and recurrence, and this is data that we got from deep proteomic sequencing of matched primary versus recurrent samples from GBM patients. And to walk you through a little bit through this busy slide, basically blue means that the protein is downregulated, and yellow or orange means that the pathway is, or the protein is upregulated. And as you can see here, I want you to focus on the recurrent glioblastoma, and you can see here that all the migration pathways are upregulated in recurrent glioblastoma, and also there's a lot more of extracellular matrix remodeling, which goes with this hypothesis. More recently, there are different studies, especially the IVGAP glioblastoma atlas project that came out of Phoenix in 2018, that defined GBM and its transcriptional and proteomic profiles based on different areas of the tumors, meaning that the cells that are in the middle, close to vessels or at the leading edge of the tumors could have different characteristics at a transcriptomic and a proteomic level. So with that, we wanted to take these results into consideration with all the studies that we do at the laboratory, and we came up with this question. First, are the cells from the tumor core and the tumor margin intrinsically different in terms of their invasive phenotypes? And we decided to go with these two areas because the tumor core is what surgeons can go in and take out, and the tumor margin is basically what is close to what could be left behind. Or if the cells from the margin are described to migrate more just because they are in contact with the surrounding cytoarchitecture. And as a spoiler for the rest of my talk, it is both. So our first aim was to determine the invasion phenotypes between these two areas. And for that, we went back to the OR with Dr. Q and Dr. Chaichana and asked them to give us 2 samples per patient based on the neuronavigation system. And they were very kind to give us samples from the core, not specifically the necrotic center of the tumor, but as you can see here in the MRI below, basically where there's a bunch of cells, not in the necrotic center. And also within the margin. The margin was taken from the enhancing areas and also with their expertise inside the OR. So far, we have been able to obtain 12 cell lines, 24 cell lines from 12 patients from the tumor core and the tumor margin, and we grew them in cell culture. We started doing some invasion assays in terms of migration, and as you can see here, this is a transwall migration assay in which we basically seat the cells in a chamber and we give them some chemoattractants to migrate. And consistently, the cells from the margin showed increased migration among all the patient samples that we were getting. This is another one that is basically the same principle, but we can measure the cells real time. And we did this for 48 hours, and although some of them are non-significant, we can see a trend that all the cells from the margin are migrating more than the cells from the core. Then we gave them a more accurate extracellular physical matrix. So we seated the cells in this nanopattern surface, which is a quasi-3D structure that cells can use to migrate more as if they were in the brain. So after 12 hours of videotaping the cells, we can see that the cells from the margin have a significantly higher velocity. They go farther, and also their directionality is more random. So with this, we can conclude that the cells from the margin have more migration characteristics. We didn't find anything in terms of the proliferation, that's why I didn't include it here. But we thought that the invasion was very interesting. We also did some studies in vivo, and as you can see here, doing imaging of the tumors over time, we didn't see any differences in growth. But if you go down to the histology of the samples, we see that the tumor core, the cells, or the tumor was more compacted and more round, and the cells from the margin were more infiltrating. They were going towards the corpus callosum. We look at these slides with the neuropathologists, and they had a more diffuse characteristic that was consistent in all the animals. We also wanted to look at the nanomechanical properties of the cells from the margin and the core, and for that we use atomic force microscopy, in which basically each one of the cells we stimulated with a probe, and after stimulation we can look at different nanomechanical properties. And we found that consistently all the cells from the margin were less stiff than the core, and they were also more deformable, which would make sense with what we were getting in the migration. And then the adhesion is kind of tricky because it depends on where they're going, they're moving, in which stage of movement and where we're taking the measurement. So we didn't get anything, consistent results, but we weren't very worried about it. After looking at the nanomechanical properties, we also wanted to see the cytoskeletal components, and we stained them for the actin cytoskeleton, intermediate filaments, microtubules. And you can see here that the cells from the margin, and it was consistent between 4 different patients, showed higher GFAP, beta-tubulin, nestin and vimentin, meaning that their cytoskeletal components were stronger, were more present in the cells from the margin. But what was interesting is that even after taking the cells out of their native microenvironment, and even after multiple passages, the cells from the margin were still showing a lot more migration in our hands. So our hypothesis is why was that the margin cells are conditioned to migrate more. So to that, we went to literature, started reading about the different components in which the cells from the margin and the core could be exposed to, meaning the ECM, the edema, other central nervous system cells. The core could be exposed to hypoxia, necrosis. So after reading and going through all of these components, we came up across the term mechanical memory or mechanical conditioning, which is an epigenetic mechanism in which external biophysical cues, meaning the extracellular matrix, can be mechanically transmitted via cytoskeletal tension into the nucleus that impacts gene expression, and that causes permanent cellular changes, as you can see here in the cartoon made by Rachel, in which we're working on a review about this mechanical conditioning in GBM. And basically the conclusion of mechanical memory is that with increased tissue stiffness, there's more cancer invasion. So now we wanted to see what is the stiffness in glioblastoma. And there are different papers that talk about stiffness measured by MR elastography, in which they describe that GBM stiffness going from the core to the margin is lower. So in the core, we have about 0.2 kilopascals, and when we were moving more towards the margin, the stiffness starts to increase, going from 1 kilopascal up to 4 kilopascals, as you can see here, in this cartoon. And very interestingly, in our brains, the stiffer areas are the corpus callosum and the white matter tracts, which are the areas that are described to be the GBM's favorite areas for migration. So we decided to go this route and study mechanical memory. So we are starting right now to condition our cells in physiologically relevant extracellular matrices. And this cell line, GBM1A, is a cell line that we don't know where it came from, in which area of the brain, of the tumor. And we conditioned this cell line for 14 days in vitro, and then we decided to study the same things that we did in our margin and core samples. As you can see here, just by culturing them for 2 weeks, we can see that the cells that are in a core physiologically stiffness area of 0.2 kilopascals, the cells are very round and not really extending. And as we increase the stiffness, the cells start to change their morphology to extend and really attach and adhere to the plate. After conditioning the cells for 2 weeks, we did a transwall migration assay, and this is work in progress. We're getting more and more results. And as we see that as we increase the stiffness, the cells start to migrate more. We also did put them in different relevant stiffnesses, the cells from the core and the margin. And we saw that when we're putting them at a core physiologic stiffness, the cells from the core migrate more. And as we're putting them in higher stiffnesses that are more relevant to the ECM of the cells from the margin, they consistently migrate more. And you can see that in the displacement graphs as well. Then we wanted to identify the main drivers of mechanical memory. And these are them based on the literature on other cancers. And we found out that YAP is one of the main drivers of mechanical memory. YAP, or the yes-associated protein, is an oncoprotein that is located in the cytoplasm and in an inactive form. And when it is activated, it translocates to the nucleus and activate transcription genes that are specific for migration, proliferation, and apoptosis. And very interestingly, Dr. Q has a lot of experience working with YAP. And as you can see here, a paper published in 2017 by our group showed that non-cancer cortex didn't have a lot of YAP protein, and then patient-derived primary GBM tissue from the samples from Hopkins had a lot more YAP protein levels. And then when we knock down YAP, we don't see a lot of migration compared to cells that do have YAP. And then you can see here as well. And then when we overexpress YAP, there is a lot more migration. So we wanted to look at the YAP expression in our margin versus core cells, and we found that our cells from the margin have a lot more YAP protein expression than the core. And we also saw that the cells from the margin, they had YAP that was already translocated into the nucleus. So then how can we target, how can we go back to the clinic, and how can we target mechanical memory in the cells from the tumor margin? There's this drug, it is an FDA-approved drug that is called verteporfin. It is approved for macular degeneration in ophthalmology. It has been used by others in glioblastoma to target invasion. This is a paper out of Mount Sinai in New York in which they use verteporfin, and they saw a lot less migration and an increase in survival in their mice. And we also use verteporfin in our lab for other types of cancer, like chordomas and metastatic brain and breast cancer to the spine with Dr. Akinduro. And we have microparticles of this drug. And one of the advantages is that it's already FDA approved. So we wanted to look at the sensitivity to YAP from our cells from the core and the margin, and also our conditioned cells. And we can see here by the numbers that the cells from the margin are a lot more sensitive than the cells from the core to verteporfin, as well as the cells that are conditioned in increased migration, in increased extracellular matrix stiffnesses. However, the problem is that verteporfin has a very big molecular weight, and it does not cross the blood-brain barrier. So right now, what we did with our colleagues in drug discovery with Dr. Caulfield and his postdoc fellows is to use artificial intelligence to come up with, to identify the most active part of verteporfin that binds to YAP, and to come up with different ligands to get a smaller compound that crosses the blood-brain barrier. So last week they shipped the compounds that we identified, and we're going to study 35 different compounds to target this protein. They're sending them our way. So hopefully we one day can translate to the clinic this drug and help a lot of patients. So in conclusion, GBM cells from the core and the margin have different biological behaviors that have to be taken into consideration when we're treating these patients. We are seeing that cells from the margin are conditioned to have increased migratory properties, and that these properties are primed by the surrounding extracellular matrix stiffness. That medical treatment in the future for GBM should be focused on margin-specific targets or the core for the tumors that cannot be removed surgically. And this is very important as well, that sampling for diagnostic sequencing should be obtained from or closer to the tumor margin or to what is left behind to get a more accurate diagnosis. And I wanted to thank you, the whole team here at Mayo, also at Hopkins. They're helping us with the epigenetic sequencing and different laboratories in the cancer centers. Thank you very much. I don't know if anybody has questions. Thank you.
Good morning, everyone. It's hard to speak after a great presentation. Congratulations, Paola. Thank you everyone for being here and everybody who participates in this work. Today I'm going to be presenting a preview of our current research we do on the immune side of the study of GBM and the fight against brain cancer. These are our disclosures. We're still waiting for a patent number before we submit any work, just as a disclosure. I would like to acknowledge the people on the screen and people that cannot be here, Dr. Q, Dr. Chin, Dr. Luo, Dr. Ulloa, as well as Mew, Yachin, Vanessa, and Harsh, who have been instrumental in this work. I always like to put this as my first slide when we talk about this type of work. And it's a quote, a very famous quote, and strategy without tactics is the slowest route to victory, and tactics without strategy is the noise before defeat. And that's a role we always talk about in the lab, especially in this project. A preview of what is CAR T-cell therapy today. As many of you might know, CAR T-cell therapy are engineered cells that express a chimeric antigen receptor against a specific tumor antigen, which can be different depending on the CAR T that you have designed. That allows for the identification and elimination of these cancer cells. In today's scientific world, the CAR T-cell therapy represents a promising avenue, which is yet to be studied and delivered for the brain, but it has been very promising against other malignancies. How is a CAR T made? Of course, you have to design depending on the generation of the CAR T that you want to use. We did not put our design here because of the patent that it's still pending. But in general terms, you have to have, of course, a fusion of the recognition domain, which is the target domain that recognitions the target antigen, the single chain domain, as well as a T-cell stimulatory domain, as you can see in the image here. More recent designs of CAR T-cell therapy now use co-stimulatory domains, which we will talk about in the next couple of slides. And how do you get the CAR T construct in the cells that you want to transfect or transduce? Well, you can use a viral vector or a plasmid or an mRNA. In the case of our team, Dr. Chin's and Dr. Luo's team designed a plasmid that goes directly into the cancer cells and transduce and transfects these cells. As you can see in the image here, for example, this is a lentivirus that goes inside the T cell and integrates with the viral DNA, and then it expresses the CAR, as you can see here. This would be the transmembrane domain, this would be the co-stimulatory domain, and this would be the target antigen, just as an example. The third generation of CAR T cells have co-stimulatory domains, and this will be important in Dr. Chin's most recent design, and this can be such as CD28, OX40, and these are all added to improve T cell response. Once these CAR Ts that are now engineered are activated, these can activate multiple immune cells and secrete cytokines and promote cell trafficking that actually kills the tumor cells. How does the T cell kills then? Well, after it recognizes the antigen, as you can see here in red, the CAR T utilizes the granzymes and the perforins axis for an effective and specific lysis of the antigen-positive tumor cells. And you can measure that in vitro, as I'm going to show you in our data. Background as of today, clinically, as we said, we have 4 CAR T cell generations over time. The most recent generations can now implement bispecific or bicistronic tumor antigen designs. It has shown great success in hematological cancers such as chronic lymphoblastic leukemia, acute lymphoblastic leukemia, as well as recurrent lymphoma and prostate cancer. And what about others? When we look at other cancers such as solid tumors, that is a big question. What is impeding these designs to target these solid tumors as well as effective as they do today with these hematological cancers? Well, we have several tumor antigens that can be expressed on different tumor cells, and they have been studied and evaluated in preclinical and clinical CAR T studies. This is a great illustration that we found, and for example, we have EGFRB3, which we know is present in 50% of tumors, of GBM tumors. Going back to the slide, we also have PD-L1, we have V7H3, and each design of each team changes the specific tumor antigen that you want to target. How are we on solid tumors today? Well, CAR T-cell therapy trials for solid tumors have yielded some good results, specifically in neuroblastoma. There's a study targeting GT2, and they found remission in 3 out of 11 patients. In sarcoma, they have found that a CAR that's targeted to HER2 has found remission in 4 out of 17, and in lung cancer, 2 out of 11 have remitted. What are the challenges of these solid tumors that is impeding for us to go forward? Number one is identifying a uniformly expressed tumor antigen, which is, I think, the most important part. That's why the efforts of having CARs that can target not just only one tumor antigen, but more, it's essential in the study of immunotherapy. CAR T-cell trafficking from blood to solid tumor sites. Of course, in case of the brain, we have the blood-brain barrier, which is something that the designs have to go through. Stromal infiltration, the tumor antigen loss, which is very important because if you design a CAR that, let's say, targets PD-L1, but the patient's PD-L1 stop expressing after the therapy, it's not longer going to be in use. And that is something that we're studying in the lab right now, which we'll show you. Tumor heterogeneity is also a very important factor we have to overcome, as well as the immunosuppressive tumor microenvironment, which Paula mentioned in her presentation. We all know that glioblastoma has a median overall survival of 18 months with surgery, radiotherapy, and chemotherapy, and cancer immunotherapy is thought to be the next way of attacking this malignancy. Immune checkpoint blockade therapies have revolutionized oncology, but have only benefited a subset of antigenic tumor types. Talking about CAR T-cell therapy for GBM, we know that, for example, 40% of all newly diagnosed patients have EGFR3 gene amplification, and about 50% of those have oncogenic EGFRB3 expressed. Therefore, if you create a CAR that expresses EGFRB3 and PD-L1, for example, can target 2 antigens and be double time as effective. And that's what we're studying right now with our design. Our current research, we are taking patient blood peripherally before the surgery. Then the team generates these CAR T cells. This is the design. We are designing a CAR T that will look for PD-L1 as well as EGFRB3 bispecifically. And then after the generation of these CAR Ts, we are testing the effectiveness and the safety after the in vitro studies, which we will show you in vivo studies to see how it behaves and the effectiveness of this therapy in vivo models. These are I want to focus the attention on CD107, which is a granule membrane cytotoxic granule component, which we can use to study how effective the therapy is at the moment in vitro. Because, of course, we know that CD107 is part of the membrane, which is membrane of the cytotoxic granule that once activated moves to the cell membrane and degranulates, liberating these perforin granzymes and things that kill the tumor. And if you measure CD107, you can measure the activated T cells in vitro. This is results of a degranulation assay of the MC999 CAR T model against the PD-L1 expression either overexpressed on the left side versus knockout on the right side. The rows below are the ones with the MC999 CAR T, and as you can see, measuring the CD107, which is the marker we were talking about of the cell membrane cytotoxicity, we can see a 33.4% of degranulation assay compared to the knockout of PD-L1. And just as context, for these studies, we used LN229, which is a commercial cell line that is very widely used in immunotherapy. But it came from a patient, from a 60-year-old female with glioblastoma. And on figure B, we can see that the LN229 PD-L1 overexpressed CAR showed more presence of granzymes compared to the PD-L1 knockout, showing the effectiveness of this construct. On the impedance killing assay, when you add a cytotoxic inducer and you put in the gold biosensor and the target cells, you can actually measure the toxicity of the inducer you're measuring. And when we did this, and the team did this with the MC999 CAR T, PD-L1 overexpressed, you can see that after the induction of the therapy, you can see the decrease in the cell index in vitro. This figure, we want to show that previous research from our team did this experiment where they saw from our cell lines the expression of PD-L1 to see which one they can use. For example, if you can see QNS 120, QNS 712 have high expression of PD-L1 in vitro, which means that they can use it for experiments because you do not want to study the PD-L1 and the CAR T effects on a cell line that does not express PD-L1. And there are some cell lines, for example, 108 that also expresses PD-L1, but in a less measure, for example, here in the middle. And when the team did the granulation assays, you can see on the bottom half row, the CAR T cells show higher degranulation assays of CD107, as measured here in the PD-L1 overexpressed versus the knockout. And it did very well with the QNS 120, QNS 612, and QNS 712. At the moment, we are using, for the experiments in vivo, we're using 108 right here because it has a more bulky growth in vivo that we have been studying at. You can measure more the mortality in vivo. These are the pre-op MRIs, T1 contrast of the patients that, for example, is QNS 120 and QNS 712, and they were both GBMs newly diagnosed. The in vivos, of course, the original experiment is we take tissue from the OR of the newly diagnosed patients. The experiment now, we're taking either low grades and high grades as well as primary and recurrent GBMs. And in between the surgery and the in vivos, we also take the blood from the patient to create this CAR T cell therapy. And then we do the in vivo surgery in the mice. So we first implant the tumor cells. For example, in the previous in vivos, we did LN229. In this case, we're now using 108, which is a patient-derived cell line. And we inject a special quantity of cells inside the mice's brain, specifically in this striatum. And we let the mice for a couple of weeks, where we then do IVIS imaging to measure the quantity of tumor cells in the brain, both in image and quantitatively. And after that, we deliver the CAR T treatment that was designed from the tumor itself and from the blood of the patient. This is the in vivo for the intracranial delivery of tumor cells and the delivery intravenously of the CAR T MC999, which was the first in vivo we did on the LN229 model. And as you can see on the row on the left, we have the group with the non-CAR Ts, which we only put inside the tumor, as well as some T cells. In the cage 2, we have the intravenously delivered CAR T cell therapy, and on the row on the right, which is cage 4, we have the PBS control groups. And we measure the tumor challenge doing IVIS every week. And with this, we want to show that the mice did persist tumor on the tumor challenge on day 14. And on day 14, this group, cage 2, actually got the liver treatment. And after delivery of the treatment, you can see that the mice on the cage 2 have non-tumor whatsoever on image and low tumor on the measure of quantity. And this stayed over day 150 where we ended the experiment. And on the figure on the right, we can see the Kaplan-Meier survival rate, where you can see the line on green is the cage 2 with the CAR-T treatment. Of course, we also then went ahead and did intracranial delivery of the tumor with intracranial delivery of the CAR-T cell to show that we showed that on intravenous delivery, the CAR-T was effective and safe in the in vivo models. Now we wanted to show that it was also efficient in the intracranial delivery of the treatment. So after putting the tumor inside the mice, 2 or 3 weeks later, we put the treatment also intracranially inside the mice, and we showed same results. As you can see, cage 1 and 2 were the ones that had treatment, and of course this is an ongoing experiment, but the tumor are alive and without tumor recurrence, as you can see in the middle here, compared to the cage 3 and 4, which has non-CAR, and they have presence of tumor, and cage 5 and 6. And these groups, the mice are dying, and in this group, the mice are perfectly fine. These are side by side, the IV and the IC, where we show the same results, as well as the Kaplan-Meier. On terms of future directions, we're repeating the intracranial model, but now the second experiment to show that it can be reproduced. And we are doing the same intracranial delivery of treatment, but with our patient-derived QNS 108 cell line, which we have here on the lab. And we also are looking at Akoya stainings to see the presence of diverse presence of tumor antigens such as PD-L1, EGFRB3, and we're studying the presence of PD-L1 days after the tumors died. So in the mice that were dead after 60, 70 days, we are dissecting the brains and doing these IHCs to see if the presence of the PD-L1 expression was still there. I don't know if anybody has any questions, but that would be my talk. Thank you very much. Okay, thank you. If there's no questions, I think we can adjourn. Thank you very much for coming, and see you next Monday.